We made this video portraying the contribution of our research project on the topic applying machine learning techniques for microbial DNA classification.
Did you know that approximately 1% of our body weight is contributed by bacteria, living on and inside the body? Yeah. That shows the dependence we have on these microorganisms, either in a good way or a bad way.
Studying bacteria came into focus in the 17th century. In 1590, Hans and Zacharias Janssen invented the microscope. It allowed people to wander about the miniature world of bacteria, making the birth of microbiology. Then they needed things to inspect from microscope.
Petri dish-based cultivation in controlled environments, commonly known as lab culturing, has been the main method of obtaining experimental bacterial samples for centuries. But the main problem with it is that only 2% out of all identified bacteria species can be cultivated this way. Yes. You heard it correct. Just 2%. Studying about only 2% is not enough.
Then the metagenomic procedure was introduced, in the late 20th century. It allowed the scientists to extract microbic genetic samples directly from the environment. These samples contained genetic information from millions of different species. Therefore, the necessity for classifying DNA sequences from such a mixture arose.
When we focus on the anatomical aspect, there are two primary sources of genetic material in a bacterial cell: chromosomes and plasmids. The big one in the middle is the chromosome. It is very, very, very large compared to several plasmids around. Chromosomes are the parts storing and preserving hereditary information. That is how you and me got some characteristics from our parent. Bacteria too pass those along new generations. On the other hand, plasmids contribute more for the survival of the microbe.
Now that you have a good understanding of the plasmids and chromosomes, let's look at the problem that we are trying to solve. The main problem is how we can classify DNA sequences into plasmids and chromosome. You would ask, why would anyone want to know if some random DNA sequence is a plasmid or a chromosome?
Glad you asked. Identification of plasmids and chromosomes is extremely important in medical research. Plasmids are responsible for the antibiotic resistance of bacteria. Yeah. They are the reason why you have to change your antibiotics so frequently. They also provide the ability to adapt with the environment.
Do you remember the nitrogen cycle? That is essential for our entire existence. Nitrogen cycle converts inert nitrogen in the atmosphere to a state more usable for living organisms. Plasmids are also responsible for nitrogen fixing, which is a crucial part of that. The few examples of numerous use cases of identifying plasmids and chromosomes separately.
Like everything in science, the plasmid chromosome classification too has come a very long way. In the beginning, scientists extracted the DNA samples from the environment and used laboratory techniques to separate them. However, this was an expensive, laborious process. Even worse, error-prone. Then comes the computational methods of plasmid chromosomes.
Okay, before getting into them, let me explain to you what a kmer is. A kmer is a unique DNA subsequence of length k. For example, AAA, AAC, AAG, AAT are 4 different kmers. Kmer frequency is the percentage of occurrence of a given kmer out of all kmers in a given DNA sequence. I don't intend to bore you with all the details of how computational methods evolved. In summary, there are 2 approaches used right now.
The first method is training machine learning models with kmer frequencies as features. The kmer frequency of each sequence is fed into a model that predicts if the sequence is a plasmid or a chromosome.
The second method is using biological features available in DNA sequences to classify them as plasmids and chromosomes. Let me give an example. In plasmids, there is a region called origin of transfer. This helps to transfer genetic information among the members of the same generation. Chromosomal DNA does not possess this. Hence, if we can identify a DNA sequence that can be an origin of transfer, it is more likely to be a plasmid.
Now let's discuss the methodology we introduced. Our finalized methodology has two main phases. We name them Metagenomics and MetaGraph. Why do we need two phases? Meta-PCBin is a plasmid chromosome classifier based on an artificial neural network model and random forest model. MetaGraph is a tool that can use to enhance the plasmid chromosome classification using graph neural networks.
Metagenomics is based on two machine learning models. The first one is an artificial neural network that uses kmer counts of DNA sequences as the model features. The second one is a random forest machine learning model which uses biomarkers as the model features.
This shows the high-level view of the user journey of the system. The user inputs a DNA sequence and the system returns the plasmid chromosome classification.
In the kmer count model, the long sequences are split into subsequences of 5,000 and the average of each subsequence probability is used to calculate the plasmid probability of the sequence. Biomarkers are fed into the random forest model which computes the plasmid probability of each sequence. A separate logistic regression model combines both predictions.
Graph neural networks are the basis of MetaGraph. They are a type of neural network designed to operate on graph data structures.
MetaGraph uses node classification as the graph neural network technique. In node classification, every vertex in the graph is associated with a label. This label is used to get the ground truth classification class. In node classification, graph nodes with higher confidence values used to train the graph. Then the trained graph will use to predict labels for the lower confidence nodes.
Our solutions surpassed the previous tools when it comes to experimental results. Both Metagenomics and MetaGraph were able to achieve the intended level of accuracy and on other metrics as well. We hope you will be able to further take our attempts further and improve on our tools as well.